Hi children, welcome back to the biology class, chapter 6, Molecular Basis of Inheritance, part 4. In this portion, we are going to explain the Alfred Hershey and Martha Chase experiment. They proved that DNA is the genetic material. In 1952, they worked with the viruses that infect bacteria called bacteriophages. Bacteria contains the protein and one of the genetic material. It may be DNA or RNA. The bacteriophage attached to the bacteria and its genetic material then enters the bacterial cell. Okay, the bacterial cell treats the viral genetic material, that it may be DNA or RNA, as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. So they started with some experiments. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein, because DNA contains the phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contained radioactive protein but not radioactive protein. Grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain the sulfur. Now again, radioactive phages were allowed to attach to Escherichia coli bacteria. Then as the infection proceeded, first stage, the viral cords were removed from the bacteria by agitating them in a blender. Second stage, blending. The virus's particles were separated from the bacteria by spinning them in a centrifuge. Third stage, centrifugation. Infection, blending, centrifugation. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria. So this one is a very important thing. See that one, bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the genetic material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that the proteins did not enter the bacteria from the viruses. DNA is therefore genetic material that is passed from virus to bacteria. This was proved by Alfred Hershey and Martha Chase. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was result from the Hershey's experiment. It became an established fact that it is DNA that acts as genetic material. However, it subsequently became clear that in some viruses, RNA is the genetic material. Answer to some of the questions such as why DNA is predominant genetic material whereas RNA performs dynamic functions of messenger, adapter and has to be found from the differences between chemical structures of the two nucleic acid molecules. Now we should discuss about what are the characters of a genetic material. A molecule that can act as a genetic material must fulfill the following criteria. What are the importance of a genetic material? What are the characters? What are the properties of a genetic material? The first property, the genetic material should be able to generate its replica. You know that replication means producing their exact copies. DNA has two strands. One is 3' to 5' and the other one it is 5' to 3' and also have the base pairing. Purine bases are paired by the pyrimidine bases by hydrogen bonds. That is called base pairing. Adenine paired by the thymine. That are the purine bases is adenine and guanine. Pyrimidine bases are the thymine and cytosine. In that, adenine paired by thymine by 2 hydrogen bonds, guanine paired by the cytosine by 3 hydrogen bonds. If we know one strand, easily we can find out the another strand of the DNA because of the complementarity. This property is present only in DNA and the RNA. These are both the nucleic acids. This ability to direct to find out their replication. The other molecules in the living system such as proteins fail to fulfill the first character. They cannot replication is absent than in the proteins. Only it is present in the DNA or the RNA. The second character is genetic material should chemically and structurally be stable. Okay, the genetic material should be stable enough not to change with the different stages of life cycle, age, or with the change in physiology of the organism. Stability is one of the properties of genetic material that we are already studied in the Griffith transforming principle itself. When we are heated the DNA which kill the bacteria, but that does not destroy the some properties of the genetic material. This now can easily be explained in light of the DNA that are two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, OH group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. RNA is also known to be a catalytic, hence it is reactive. Therefore, DNA is chemically less reactive but structurally more stable. See this one, DNA is chemically less reactive but structurally more stable when compared to RNA. Among the nucleic acids, DNA or RNA, in that DNA is a better genetic material because DNA is structurally more stable than the RNA. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Next point, the third point is it should be the genetic material should provide the scope for slow changes, that is called mutation, that are required for evolution. Both DNA and RNA are able to mutate. Mutation is present in both the cases. In fact, RNA being unstable mutate at a faster rate because viruses having RNA genome and having shorter lifespan mutate and evolve faster. Because of that reason, DNA and RNA, they can represent the changes, but compared to that, RNA is fast. Our next point, it should be able to express itself in the form of Mendelian character. RNA can directly code for synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? Okay, RNA was the first genetic material. There is now enough evidence to suggest that essential life processes like metabolism, translation, splicing evolved around RNA. So RNA is used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzymes. But RNA being a catalyst was reactive and hence it is unstable. See, listen, RNA being a catalyst were reactive, so it is unstable. Therefore, DNA was involved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strand further resist changes by evolving a process of repair. Thank you.